and that is Dr. Sarah Kraus. And she is going to present the Sarah nanotechnology. Where is that name coming from? Sarah. Very nice. We look forward.
So, good afternoon. I'll be starting with a brief introduction on Sarah nanotechnology, which was designated as a medical device that comprises Sarah nanoparticles and an electromagnetic induction system. Sarah nanotechnology is aimed primarily for the treatment of stage 4 small cell lung cancer, but we are also studying potential treatment applications for other cancer types. Now, the Sarah nanoparticles are given intravenously to the patient, and they become localized on the cancer cells through the use of the EPR effect. Although this is a systemic treatment, there are no side effects. Now, the main innovation of the Sarah nanoparticle relies on its ability to control its temperature without inducing thermal ablation. Now, the model of choice in the experiments that I will show you today was based on the 41 triple-negative mammary carcinoma that forms spontaneous metastases in the lungs and is similar to those found in human breast cancer. So this is the Sarah nanoparticle that is composed of 3 main components. You have iron oxide nanoparticles that heat under the application of an electromagnetic field. We have a phase change material, or PCM, that can absorb the energy without heating above its enthalpy point, and therefore limits the temperature of the Sarah nanoparticle to 50 degrees. We also have an encapsulating polymer, this is SPEC 20,000, that encapsulates both the PCM and the iron oxide nanoparticles, thereby enabling flexibility, as you can see here, durability and biocompatibility, and also masks the Sarah nanoparticle from the macrophage and phagocyte system. So here you can see an image captured by transmission electron microscopy. The overall size of the Sarah nanoparticle is 135 nanometer. This is the electromagnetic induction system that is composed of 2 main units. We have a cooling device, that cools the system, and an RF generator that generates the electromagnetic field. Now we put the animals inside a housing tube, and then this tube is positioned inside the inductor coil for EMF application. So I'm going to show you a series of experiments that we have conducted at our company. And the first one is the biodistribution in mice, aimed to evaluate the distribution of Sarah nanoparticle in vital organs and blood, and particularly in tumor tissue over time. And we used the 41 breast cancer metastatic model, and the mice received a single injection of Sarah nanoparticle, and then they were sacrificed 2, 4, and 8 hours after the injection. Then we quantified the amount of Sarah nanoparticles by a squid that measures the magnetic properties of the nanoparticles. So you can see here the accumulation of Sarah nanoparticle in the lungs, and, which was the highest at 8 hours post-injection. And here you can see the distribution in blood, which was the highest 4 hours post-injection, and decreased thereafter. So there is a trade-off between the blood and the lungs, and when the amount in the blood decreases, it allows the amount in the lungs to increase. Now, the amount of Sarah nanoparticle that reaches the lungs is quite high. It's about 20%. This is compared to other studies that have shown that only a small percentage, less than 5%, ends up in the target organ. So we have quite a lot of nanomaterial in the lungs. We have conducted toxicity evaluation in mice. We used healthy biopsy mice that were treated with 1 cycle of SARA nanotechnology treatment that included a SARA nanoparticle injection, followed by 3 minutes of EMF application at 5 hours post-injection. Then we sacrificed the mice at 3, 14, and 30 days after the treatment. Now, to make a long story short, we found no mortality among the animals. There were no abnormal clinical signs in any of the animals throughout the observation periods. All animals exhibited a normal body weight gain, and histopathology evaluation did not find any treatment-related changes. We only saw some sporadic cases of pigment-laden macrophages in the liver and in the lungs of the treated animals. Now, these changes were minimal, and therefore they were not considered as adverse. And here you can see some representative photos of H&E stainings in the lungs and in the liver. And you can see here the green arrows here indicate small collection of pigment-laden macrophages in the lungs that were associated with minimal mononuclear cell infiltration. Same situation in the liver. These are Kupffer cells, only minimal changes in the macrophages. We also evaluated the efficacy of treatment in the 41 breast cancer model. The animals here received 3 cycles of SARA nanotechnology treatment. In each treatment cycle was given within 2 days intervals. The primary endpoint was the number of lung metastases as determined by a visual counting of the metastases as well as ex vivo imaging of the lungs. We expressed an M-cherry fluorescent protein in the cancer cells, and the main findings of the study showed almost a 50% reduction in the number of lung metastases, a 70% reduction in the relative size of the largest metastatic nodule in the lungs. This was done by morphometric analysis by histopathology evaluation, and more than 90% reduction in the fluorescent intensity of the metastases, as done by the ex vivo imaging. So here you can see the data. These are the results of the macrovisual count. You can see that the number of metastases in the treatment mice was significantly reduced. These are the results of the ex vivo imaging, M-cherry metastasis, the heat map of the viable metastasis, as well as the quantitation that was plotted here. And you can see that the total fluorescence signal in the lungs of the treated mice was significantly lower, supporting both a reduction in size and in viability. Our last study was a survival pilot study in the 41 M-cherry breast cancer model. This time we gave the animals 5 treatment cycles, and the mice were followed up until the last mouse died, and we recorded all the dates of death. And you can see here the effect of treatment. The effect of treatment on the median survival time, which was 21.6 days in the control group and 29 days in the treatment group. And here you can see the Kaplan-Meier survival curve, showing that the percentage of survival in the treated mice was higher than that of the control. And at day 33, 40% of the animals in the treated group were still alive, while all the mice in the control group have died. Therefore, we're able to prolong the survival of the mice by a third. To conclude, systemic administration of SARA nanoparticle followed by EMF application was not associated with any adverse reactions. The results of the efficacy study showed a significant reduction in the number of metastases as well as in the size and viability of the metastases in the treated mice. And also the survival of the treatment mice was significantly improved compared to the untreated control mice. So where are we heading now? We are currently working on the production of SARA nanoparticles under sterile conditions, and we have built a clean room for this purpose, which is now fully operating. We have started GLP studies in mice, and we are soon to start GLP studies in swine as well. And the first in human clinical trial is to be started in North America, hopefully next year. I would like to thank all the people involved in these studies, and particularly Imagen Biosystems in the US, also to the organizers of the summit for the opportunity to present our data. And here you can see a picture of our newly inaugurated clean room, and thank you all for your attention.
Thank you for showing us an alternative way of treating cancer in this conference. I think you're the only one with iron oxide, at least what I've seen, but maybe more were there. Questions? Your Sarah, it's probably you were the inventor?
No, actually I'm not.
So you're not related to the technology or your name?
No. No.
Oh, that's a coincidence.
By coincidence, yes.
Okay. These SARA nanoparticles, they contain iron oxide nanoparticles in one particle.
Yes.
So how big is it? What's the
I
size?
showed it in the same image. It was 135 nanometers.
135. In the beginning of our session, we had the clinical results of hypothermia,
Right.
which shows sort of prolonged survival. It's not strong. What are your aims to reach in what type of tumors with this approach?
Well, it's aimed primarily for the treatment of stage 4 small cell lung cancer. All these experiments were done in the 41 model. This is triple negative breast cancer. But of course, we're looking at additional solid tumors in which we can test technology in terms of efficacy, of course, survival, and so on.
But if you, I see the end, but if you overlook the literature and clinic, cancer, hypothermia, what is actually the best you can get with hypothermia in terms of treatment response? If you oversee the whole literature, is it a couple of months more in survival, or?
We're talking about stage 4 patients, and we're aiming to do sort of a chronic treatment to prolong survival, and
Okay, thank you. Then.
Yes, a technical question. So, how many iron oxide particles are within one particle, and how robustically you can make them? So let's say you have 10 per particle, okay? Can you repeat this number, or it's not important?
I think it's not important. What we need is, there is a tight association between the amount of nanomaterial that reaches the tumor and the efficacy of treatment. So what we need is to have a thermic mass in order to have an effect. So the results is pretty reproducible. We have repeated the experiment several times, and we get the same results. I think it's not that important. Of course, for for the human trials, we will have more. We will need more standards, but I think it's not that important.
Thanks.
So to follow up on these approaches, what we hear also today is that it's very important to intervene with the immune response and with all of these checkpoint inhibitors and all of these pathways that they are suppressing the tumors. Are you not thinking to combine the two approaches with the delivery of maybe an antibody to any of these molecules, or to target maybe a function on the tumor or on the T cells?
At the moment, no, we're not thinking on combining antibodies.
It's just thinking that you could generate, maybe not only eliminate part of the tumor, but you will have some antigens that now they will be in an environment that is immunogenic, and if you even suppress the immune, the suppressed response, this will be...
Yes. I know, but the technology is the system, the nanoparticles and the induction system. At the moment, we're not planning to combine or any antibodies to it.
Yes, in the back.
Thank you very much for the nice talk. I didn't technically understand, do you need to know where the tumor actually is when you come to heat it up, or can you just systemically sort of use the radio frequency and wherever the nanoparticles are, there will be heating?
The nanoparticles are given IV to the mice, and the EMF application is a whole body irradiation. And the EPR effect enables the preferential retention of the nanoparticles in the tumor, but we're hitting the whole body of the animal.
So I would hypothesize that there is substantial heating of the liver, probably?
Well, according to our tox studies, we don't see any toxic effects in the liver. As I showed you before, we see only minimal infiltration to Kupffer cells on the resident macrophages in the liver. And we also, of course, when you inject nanoparticles IV, a significant amount will go to the liver. It's not something you can avoid. But we have done long PK studies, and we have followed up the mice for up to 2 months. And the nanoparticles get cleared and excreted from the body. So I guess if we wait longer, we'll see a reduction. But according to the toxic effect, we haven't seen anything. Like everything was normal.
Thank you.
You're welcome.
Okay, thank you. Thanks so much. I think we saw quite a variety of different approaches to improve cancer therapy. I keep it short a little bit because we had extensive time for discussions. And I would like to thank the presenters for their presentations, and you all for listening and the discussions. So it's break time, and maybe see you later.